Neurodegenerative diseases affect millions of people globally, with Alzheimer's and Parkinson's being the two most common conditions. Researchers and clinicians worldwide are working to understand how and why these diseases affect our brains and ways that we can prevent and treat them more effectively. Scientists hypothesize that the accumulation of metabolic waste products, like certain types of proteins and peptides, is one of the underlying causes of many neurological diseases. But it's likely that lifestyle choices, genetics, age, and other environmental factors play a significant role as well. Another issue with studying neurodegenerative diseases is that each one has its own clinical symptoms and pathology, making it difficult to find one solution for diagnosing and treating all of them. But in 2013, a team of researchers made a groundbreaking discovery about our brain's ability to remove toxic waste that might offer key insights into the study of neurodegenerative diseases. Dr. Maike Nedergaard and her colleagues from the University of Rochester Medical Center published a paper proposing that the brain has a system to remove neurotoxic waste products that accumulate in the awake central nervous system. The researchers decided to call it the glymphatic system because it involves our brain's glial cells and because it works similarly to our lymphatic system in our peripheral organs. Think of the glymphatic system like the brain's waste removal service, clearing out toxins and metabolic debris while we sleep. We all know the importance of a good night's sleep, but learning more about the glymphatic system might explain one of the reasons why sleep is so crucial for maintaining good brain health. While there's still a lot we don't know about the glymphatic system and how it works, it has gained significant traction among brain imaging researchers for its potential to study neurological disorders. One of these researchers is Dr. Nan-kuei Chen, a biomedical engineering professor and MRI physicist from the University of Arizona. Dr. Chen and his colleague, Dr. Ying-hui Chou, are working on two different methods of neuromodulation that could potentially be used to enhance brain waste clearance. The first method is transcranial magnetic stimulation, or TMS for short. Dr. Chou is the test subject and is getting an MRI to map her brain before undergoing 3 rounds of TMS. TMS is a non-invasive technology that sends painless magnetic pulses to the brain. It is an FDA-approved treatment for depression, but Dr. Chou also studies the effect of TMS on mild cognitive impairment in adults.
The way of modulating glymphatic activity through externally applied electromagnetic wave is kind of novel in some ways, so that's why it's important that once we do that, we need to use good MR technology to see if we can see any effect on glymphatic activity.
Okay. Acquire. And ear. Acquire. And the big coil, we have found a spot using basically looking at white matter tracts in the brain that we think is going to most correspond to the hippocampus, which is our sort of target area for memory. And so we'll just be doing that stimulation.
What we are trying to do is that even though we are not sleeping, even though we are not in non-REM sleep stage 3, if the externally applied EM wave could enhance the activity. So maybe that's another interventional procedure that could be applied to human subject.
After 3 rounds of TMS, Dr. Chou receives 1 more MRI in order to study if there are any effects on the glymphatic system. For the second glymphatic system study, Dr. Chen will be the one receiving neuromodulation. In this experiment, the MRI machine will not only be used to image his brain, but will also be used as a neuromodulation tool. This is a novel approach that uses an MRI machine's hardware components to produce a neuromodulation effect. Under certain uncommon conditions, MRI machines can produce harmless nerve stimulation in the form of muscle twitches.
What our idea is that instead of treating that as an undesirable effect, if we control the effect carefully following what we know about neuromodulation, there could be a way to perturb nervous system. It's safe within FDA guideline, but effect, although subtle, is there.
So could you actually feel anything when you're
I don't feel anything because this is the sub-threshold stimulation, because the gradient coil in MR hardware is actually capable of inducing a central nervous system activity if we design the product correctly. Yeah. I guess that's my brain, right?
This is Ying-hui's brain. I checked Ying-hui's brain.
Oh, Ying-hui's brain from 2 days ago.
Also,
Once Dr. Chen receives the MRI-induced neuromodulation, the results of both studies are evaluated by Dr. Qiuting Wen, an imaging researcher from the University of Indiana Medical School.
My research is dedicated to utilizing neuroimaging techniques to study a crucial component of the human brain, the cerebrospinal fluid.
Cerebrospinal fluid is a clear, colorless liquid that surrounds and cushions the brain and flows through a network including perivascular spaces. Scientists hypothesize that the glymphatic system clears neurotoxic waste through a process involving CSF and other neurofluids. MRI machines are actually able to track the flow of CSF as it moves throughout the brain.
This makes MRI a perfect fit for studying CSF dynamics and its circulation within the glymphatic system. We can find a way of improving the waste clearance. It will benefit all kinds of neurodegenerative disease. It's not specific to either Alzheimer's disease or Parkinson's disease.
After Dr. Wen evaluates the data from the two studies, the three researchers meet virtually to discuss the results and to determine whether or not these glymphatic system experiments are worth exploring further.
Comparing before and after, the most notable change to me is this peak shift. This is not very commonly seen for my reproducibility analysis.
While these results don't necessarily point to specifics about waste clearance, they do show that there were changes in both Dr. Chou's and Dr. Chen's brains.
Even though if we don't see same result from two stimulation, I think each of them is interesting enough to move forward.
I think one exciting aspect of this glymphatic system is that we can modulate. It seems it's much easier to modulate than other pathology pathways.
The next step, we might change that duration of stimulation and also interval between sessions, and also who will benefit, who will be the best population to target.
I like to join you guys and learn more and seize the opportunity to push the field forward.
While these initial results are exciting, there are still many unanswered questions, both for Dr. Chen and the field as a whole.
So who knows what we'll learn, and maybe we'll be changing our approach of tackling this question. So we'll keep our mind open and we'll see what happens. We'll follow the data.
There's still a long way to go before scientists understand the glymphatic system's full potential, but with enough time and research, it might one day lead to some significant breakthroughs in our understanding of the complex nature of the human brain.